Hi students, this is Dr. Badrinath and today's video is a targeted drug delivery system. Yeah. See, the doctors, suppose say there was a problem in this eye, suppose say there was some cancerous or infections. They will do a surgery only to the infected area where there was a problem. They will do surgery to that part only. They will not touch the remaining part. Okay, you got it? Very beautiful concept. They are very intelligent. That's why what the pharmacists also they are thinking in such a way. See, if we are giving a formulation, it is circulating throughout the body, including the healthy tissues also. Unnecessarily, drug is exposing to the healthy tissues also. Why can't we prepare the formulations in such a way that our drug, our formulation, our drug has to reach only to the problematic area. So this area, suppose say, that's the target area. Why unnecessarily we have to expose the drug molecule to the healthy tissue? You got the point? That is the logic here. See, we have to learn from the doctor. The pharmacist now they have learned from the doctor. On fine day, they got a wonderful idea. Why can't we prepare a targeted delivery system, targeted drug delivery system? See, we are not giving a drug to the healthy persons. Isn't it? We are not at all giving a tablets or capsules to the healthy persons. Isn't it? We are giving only to the patient. In the same fashion, to the patient also, why unnecessarily we have to give to the formulation, we have to expose the drug molecules to the healthy part of the body. See? Okay. From that concept, we have made the targeted drug delivery system. Keep in your, we are calling as a TDDS. TDDS, we are also calling as a transdermal drug delivery system. It is a we have a two abbreviations for TDDS. Keep in your mind, but don't get confusion in the examination. You can see the concept here. Wonderful concept of the transdermal drug delivery system. See the center person. He is the patient having a problem in his lungs. So, and we have given a medication. We have given a formulation in the form. We have given the drug in the form of a formulation. Okay, right. So what happened in case of conventional drug delivery? This drug, see the color orange drug is distributed throughout the body. But where is the problem? Problem is there in the lungs. The drug has to reach to the lungs. Okay, right. Suppose say if it is a targeted drug delivery system, this reaches directly only to the lungs. Drug molecules will reach only to the lungs only. See the color intensity. See, this is a very good bright orange color drug reaches completely into the lungs. The remaining healthy tissue remains only in the same healthy only. Right? You got my point. See here, there was a two problem with conventional drug delivery system. Unnecessarily here the drug is exposing to the body, that is to the drug. That is number one. Number two, another important problem is also there. The problematic area, lungs, is having a only less concentration of the drug because the remaining unnecessarily the drug is distributing throughout the body unnecessarily. So what happened? Only less concentration, less amount of the drug is went to the lungs. But whereas here 100% what you have given, the 100% of the drug is, see the color intensity is, it becomes a very less. Here color intensity is more, same color intensity. Here color intensity is red. Where is the remaining drug? Unnecessarily exposing to the exposed to the body. Yeah. Now you can see here other tissues. Here this is a target tissue or infected tissue, target tissue. These are all the drug molecules. See, the drug molecules will go and reach only to the target only. See, other tissues are not at all exposed to the drug. That's why we are calling it as a targeted. It go and reaches only to the target area, site of action, site of problem. Okay. See, high drug accumulation you can be found in the targeted drug. Right. Now you can see the differences in the examination. You have to write all these points and diagrams. Very, very important. Drug is distributed, same points what I have explained, the same points only here. Can be take a screenshots, Okay. Yeah, this is your syllabus. Here you can see targeted drug delivery concepts and approaches, advantages, disadvantages, all are mentioned here. Okay. The same things I have divided in this one and I covered in this targeted drug delivery system. This is my mobile app. You can be downloaded from the Google Play Store. Okay. All my presentations are available in the PDF format. It is nothing but a notes. You can be write as it is in the examination, need not to be purchased any textbook or need not to be take Xerox. Okay. And after opening the app, go to the store option. See the pointer here, store option. There I have arranged our notes in the form of your subjects as per the syllabus, Pharmacy Council of India syllabus. And this is my YouTube channel. Go to the playlist option. Here I have arranged all my videos in the subject wise. If you click on each subject, again you will get a topic wise. Yeah, now you can see introduction to drug delivery, targeted drug delivery system. It is a type of drug delivery system where the drug is delivered to a specific location, site of action, rather than the whole body. Very simple definition. Drug concentrate in the tissue of interest, tissue of problem, tissue of target, like that. Always aimed at high efficacy at the site of action. It shows the high efficacy at the site of action, no toxicity to the healthy tissues. That is the biggest advantage. No side effects to the remaining tissues, no toxicity. It is a combination of the, but see, keep in your mind, it is a combination of the diverse fields like a polymer science, pharmacology, bioconjugate chemistry, okay, and molecular biology, electric fields, magnetic field. Everything I will explain you in the slides. And if you want to prepare the drug in the form of a targeted drug delivery system, we need some carriers. Drug, if you don't give drug alone as it is, if you are giving a drug as it is without any modification, drug will be circulated or it will be traveled throughout the body. So you have to prepare in such a way that take the drug, keep in a carrier, keep in the carrier. Okay, these are all the special type of carriers. These carriers having ability to go reaches only to the problematic area, like 0D, 3D, 3-dimensional. This is 1-dimensional. It is a 0-dimensional. It is a 2-dimensional materials. Mesoporous, liposomes, micelles, hydrogel, polymeric nanoparticles, dendrimers, and here if you see the dimensional, 0-dimensional, fullerene, 1-dimensional, carbon nanotubes, 2-dimensional, graphene, 3-dimensional, graphite. All these points you have to be right in the examination along with the structures, right? Okay, carriers now are ready. You have a drug particles in your hand. Another hand, you have chosen a very good, excellent carriers. Okay, right. Now your target. Approaches. There is a number of approaches to get targeted drug delivery system to achieve. There is a classification out there. This classification, 4 types you can be see here. This is a type 1 classification, type 2 classification, type 3 classification, type 4 classification. Okay, type 5 is also there, but no problem. Totally type 5 types are there. Type 1, 2, 3, 4, 5 are there. So, but the important points only I have covered here because it is a very big assay. Okay, and type 1 classification, this is based only on the modification of the drug particle. If you are modifying a drug particle, if you are modifying a drug carrier, then you can say based on that active targeting, passive targeting. Active targeting again 4 types, so first order, second order, third order, fourth order. And based on the target nature, if your target nature, where you want to target, okay, that environment nature, if it is a biological targeting or physical targeting, chemical targeting, and based on the area of the target, you want a systemic target, you want a locally target. And here there is some other types are there: inverse targeting, dual targeting, double targeting, combination of the targeting, combination 3 or 4. And classification based on the site of action, nature of the disease. Okay, disease based target, local based target. Yeah, now we will see the important one, passive target. Here, what is the passive targeting? We are not modifying a drug. Yes, you heard it correctly. We are not modifying a drug surface. We are not modifying a carrier particles. Drug particle surface is not modified. But however, it reaches to the target site. Reaches. How? See, it reaches to the target site because of the body's natural response to a disease, inflammation, infection. Example, wonderful, very nice example is there, this concept. See, this is a cancer cells. This is a normal cells. This is a blood capillary. We know that blood capillary having a endothelial cells. These are all the endothelial cells. And these are the drug particles. See, endothelial cells of the cancer cells. See here. See, endothelial cell. That is a blood vessels. Blood vessels wall having an endothelial cell. This endothelial cell permeation is more. They have a high permeation only in the area of the cancer. Wherever there was a cancer in our body, the surrounding blood vessels, endothelial cells, it permits more, high permeability. See here. Here is a very tight junctions here, endothelial cells. Here, endothelial cells becomes looser, high permeation to the drug molecule. So automatically, it is body's nature. Whenever there was a cancer cell, automatically there was a loosening of the junctions of the endothelial cell. So drug passively, just casually it goes and it enters into the cancer cell area and it releases the drug there. This type of targeting is called a passive targeting. This is called as a enhanced permeability of endothelial cells in the area of cancer cells. This is called EPR, enhanced permeability and retention. It retains there itself. Enhanced permeability and retention. Anti-cancer agents we are using to target the anti-cancer cell. Now, active targeting. Passive targeting is over now. Active targeting. Here we are doing surface modification. Surface particle, drug surface, drug carrier surface. Keep in your mind. Here I am using both. Drug means nothing but drug carrier. Keep in your mind. Drug carrier. So for example, say liposome. Drug, anti-cancer drugs we have placed in a carrier. This carrier is nothing but a liposome. This is a temperature sensitive. Okay. Suppose say healthy environment, healthy cells. These liposomes will not become looser. The liposome structure will be tight. So no drug is released. Suppose say 37 degrees centigrade, that we know very well. In case of cancer environment, there was a little increase in the temperature. 39 to 42 degrees centigrade, here 37. So mild hyperthermia. This is cancer cell, not the egg omelette. Don't think it is a egg omelette. It is a cancer cell, man. It is very dangerous. Okay. So what happened here? The liposome we have modified in such a way that it is a thermosensitive, where there is a high temperature like 39, it will become loosened and inside it releases the drug particle into the environment. Okay. This is called a active targeting. We are doing a modifications to the carrier. Like here, heat sensitive polymer and the coating of the carrier with the ligands like a biological ligands like proteins, polysaccharides, peptides, tissues. We are giving a coating to the liposome or to the carrier. And why there was a first order, second order, third order, fourth? What is this fourth? It is not sustained release. First order release, zero order, not that type of here. In the biopharmaceutics, you will get. Here that depend upon target side. If your target side is retained only on the capillary bed, then you can say it is a first order. And if you want to deliver the drug to a specific cells, like a tumor cell, cancer cell, then you can say it is second order. Third order, drug will be delivered to the intracellular components. And fourth one, drug will be delivered into the nucleus, that is a DNA are proteins, macromolecules which are present inside the cell, inside the nucleus. Okay, so that, yeah, here I have shown this is a active targeting example. All these you have to write in the examination, my dear students. Here you can see the drug particles. See the pointer, drug particles. We have this drug particle we have placed in the carrier. See here, this is a carrier. This carrier we are attaching the some ligands. All these are the ligand. Here 3 parts you have to observe. Number 1, drug. Number 2, carrier. Number 3, ligand. See how we are designing these ligands. See, arrow mark. So I have designed in such a way that whenever I have given this particular ligand having a, see, it go and attaches to a specifically to the receptors. These receptors are present. These type of receptors are present only on the target cell. So see the exactly it fits. Okay, and it enters. So the drug will be released there. Okay, this is, see, examples for these ligands, biological ligand. The number of ligands are there. This ligand, either it may be a protein, or this may be a polysaccharide, or it may be a aptamers, or it may be a peptide, or it may be a small molecules. Okay, this is called a ligand, carrier, drug. And physical targeting. Here in the physical targeting, we are using some physical means. Keep in your mind. Physical. We will keep the magnet here to the patient body. Externally, we are placing a magnet here to the external and not inside. External environment to the patient's stomach, near the stomach where the problem is there. Where suppose say there was a cancer, we are keeping a drug magnet here, and we are giving an injector. So injectable suspension of that, see, it is a targeted one. It consists of a iron. Iron here, ligand is iron. So iron and the magnet that you know very well, drug is attached with the iron. Then that we are giving an injection. So the drug will be reaches directly to the tumor site or the cell. So we are using a physical means like a magnet. Here I have shown the magnet here. And in the same fashion, electric, we are giving some electric current here. Where there was a problem, we are giving electric current, and also we are giving a magnet. This is a magnet is over, electric current. We are giving an ultrasound, like physiotherapy. You just ask them physiotherapy. And where there was a problem, you can put some ultrasound. So what happened? You are bigger in the ultrasound, drug will be broken in the ultrasound area. Your carrier will be broken. Drug will be released outside. See here, outside. Drug will be released and it reaches to the area. And the light, see here, light. And you can put a focus on the light. Physical means, so whenever inside the drug, what happened? Whenever there was a high light, the carriers will be broken and the drug particles will be released. And dual targeting. In dual targeting, you have a 2 target, target 1 and a target 2. See, blood-brain barrier cancers, cancer which is present inside the brain. It was a problem. What is that problem? It have, see here, in dual targeting, keep in your mind, we have a 2 targets to reach the target site. Hence, carrier molecules are functionalized. Carrier, see here, this is a carrier. This carrier we are functionalized with the 2 activity. See the blue color, see the pink color. 2 targets having a 2 types of ligands. Target 1, number 1, it has to cross the BBB, blood-brain barrier. So that's a target 1. And once it is crossing the blood-brain barrier, see target 1, blood-brain barrier. Once it is crossing the blood-brain barrier, because your tumor is present inside the brain. Okay. See here, brain cancer cells having a specific marker so that our target 2. So target 1 is crossing the blood-brain barrier. Target 2 is reaching the tumor cells. So it is having a 2 targets. And double targeting. Here, what is a double target means? Drugs are targeting to a specific organ. That is number 1. Controlling release. Rate of drug delivery to the target site. Controlledly it has to release. Two objectives are here. One is targeted drug delivery and controlled drug delivery. Suppose say Both are combined. That's why this is called a double target. And inverse targeting. Normal is boring. Like that we are inversing. See this fellow, inverse. Here what we are doing is a inversion. It is very interesting. I have already told you that near the cancer cell, endothelial cell permeability is high. That is natural. We are making it reverse. Reverse we are making by giving an iron dextran injection. See, this is iron dextran injection. We are injecting to the area of the cancer cell. So this endothelial cell of the cancer will become tight. It will become tight. So drug will not go here. Inversely we are targeting. So here we have made attempts to avoid the passive uptake. Passive targeting we are avoiding here. Okay. Example is suppressed. We are suppressing the reticular endothelial system. We are suppressing here. That's why this inverse targeting we are using for non-RES organs, non-reticular endothelial system. That is a non-RES organs only we are using this inverse targeting. This example you have to be right in the examination. Okay, like that many are there. Important I have already covered. Need not to be right the remaining. Don't bother about that. And what is the need of developing this targeted drug delivery system? See, conventional limitation, conventional drug delivery system, see like this. Conventional drug delivery system is having a problem. What are that problem? It is having a pharmacokinetic problems like a half-life, high volume of distribution. It is having a problem of pharmacodynamic, low specificity, low therapeutic index, low solubility, drug, pharmaceutical factors, low soluble. Some of the drugs are low soluble, low stability, and pharmacotherapeutic problems also like a high dose, adverse effects, low patient compliance. All these are the problematics with the conventional drugs and conventional drug delivery system. That's why we are modifying. Okay, solution. And finally we got a solution that is for a targeted drug delivery system, and these are the advantages: less dose, high efficacy, less side effects, reduced toxicity, avoids the first pass metabolism, drug remains at a tissue for a longer time and increases. In case of infection, drug will reach only on the microorganism. Your body will not at all be exposed to the drug molecules. Healthy body, only microorganisms, bacteria, fungus which is present in your body, only those will receive the drug, not your healthy parts of the body. Disadvantages: chances of immune system reaction is one of the problem is there. It needs a high sophisticated technology. It needs a high manufacturing cost and high manufacturing time also. Very long. A patient is waiting here. They will say, I am doing a research. I am doing a research. I am doing a wait for the patient will die within the time. Okay. Difficult to maintain the stability. For example, say a resealed erythrocyte. It needs only 4 degrees centigrade. And difficult to predict and difficult to fix the some dosage regimens like morning one tablet, evening one tablet. It was somewhat difficult here. Okay, dosage regimen. Okay, and here you can see all my what is my services like YouTube and app, both